Hey, everyone, and welcome to episode 102 of Cell and Gene, the podcast. I'm Erin Harris, and you're about to listen to my conversation with BioCardia's CEO, Peter Altman, Ph.D. BioCardia is a clinical stage regenerative medicine company that develops novel therapeutics for cardiovascular diseases with large unmet medical needs. And during our conversation, Peter talks us through the company's evolution from a cardiac biotherapeutic delivery firm to a developer of autologous and allogeneic cell therapies for cardiovascular and pulmonary diseases. He talks us through the company's novel approach to treating ischemic heart failure using autologous cell therapy, and he also shares insights from their phase 3 clinical trials. He offers some really strong insight on the advantages and challenges of autologous versus allogeneic cell therapies, and what it will take for regenerative medicine to make a lasting impact in cardiovascular care. So listeners, I learned a great deal from this episode, and I really hope you do too. So with that, I'll let you go and dive into this episode. And so here's my conversation with BioCardia's Peter Altman. Enjoy. Peter, welcome to Cell and Gene, the podcast. Thanks for being here.
Thank you for having us, Erin.
Absolutely. All right. So tell us a little bit about BioCardia.
BioCardia began as a cardiac biotherapeutic delivery company. And over the years, we've evolved to developing cell and cell-derived therapies for both cardiovascular and pulmonary disease. Our platforms include our autologous CardiAMP mononuclear cell therapy, our allogeneic CardiALLO mesenchymal cell therapy, and our Helix transendocardial biotherapeutic delivery system, where it all began. Our lead program is the CardiAMP autologous cell therapy, and it's focused on the treatment of ischemic heart failure of reduced ejection fraction.
Excellent. Thank you for that description. And we will get into some of what you just discussed, specifically CardiAMP and a few others throughout the discussion. But before we do, I'd like to pick your brain a little bit about the fact that, so to date, there's no cardiac cell therapy has received FDA approval. And so I'd like to hear from your perspective, what would you say have been the biggest either scientific, clinical, and or regulatory hurdles that have been preventing this approval?
Now, this is a real big question. So I actually would distill it into the two most significant hurdles being both the scientific and clinical development, which are interrelated. Essentially, there are two general approaches today for cardiac cell therapy. That of trying to stimulate a response in the tissue from agents that the cells release locally, such as we are pursuing in our programs in CardiAMP and in CardiALLO, and those that are intending to provide cells that repopulate the myocardium with new cells. Each of these approaches has some very common challenges, such as delivery, how the cells get to the heart, cell handling, how one manipulates the cells prior and into treatment. But they also have very unique challenges to each cell type for each different indication. And some of those have been talked about greatly. The idea of an allogeneic cell being rejected by the immune system. In the heart in particular, cells intended to repopulate the heart also have the potential of triggering life-threatening arrhythmias. And that's been a challenge that folks have been working on for some time. At BioCardia, our origins, as I mentioned, lie in solving the delivery problem. And so we feel pretty comfortable there, and we've worked with others developing cell therapies using our delivery system. And the approaches we are pursuing in our cell therapies at BioCardia have not had any of the issues of rejection or causing cardiac arrhythmias. So we don't share those similar challenges and probably are not as sophisticated in talking about them as others are, because we don't have the issues of rejection and potential for life-threatening arrhythmias.
Excellent. Okay. I want to talk a little bit about CardiAMP, which we, of course, referred to at the top of the call. So CardiAMP stands out by screening patients for likely responders before treatment. Talk to us a little bit about why is this pre-procedural cell analysis so critical, and how does it improve both trial outcomes and the potential for perhaps regulatory success?
Happy to do that. So, Erin, we've long understood that the very nature of an autologous cell therapy is that there is great patient-to-patient variation in the actual therapeutic cells delivered. They're autologous. They come from that patient, and then they're processed and handled and readministered to that patient. Based on previous clinical experience, it became apparent that the source cells from the patients for the actual therapy have correlated with therapeutic outcomes. And this makes sense. You're essentially characterizing the source tissue from which you're deriving your therapy, but it can also contain additional information on the characteristics of the patient that we may or may not fully understand today. But by doing this prospectively and selecting patients with cells likely to respond to therapy as an inclusion criteria in our program, it focuses our development on those most likely to respond to the treatment. And the more responders in a target population, the smaller the trial that is needed to demonstrate benefit. So regulatory success is all about developing the evidence to support safety and benefit, and selecting patients and processing cells at point of care instead of selecting cells and processing them remotely preserves the enormous value of the autologous nature of the therapy in terms of safety for patients. And it also eliminates the problem of the cost of autologous therapy. That further supports the pathway for adoption in the clinical world. So that's sort of how we've gotten here, and we're pretty pleased with what we're seeing.
Yeah, certainly, for sure. Very interesting. So many of the cell therapies, they take a one-size-fits-all approach, but CardiAMP, tailors treatment based on individual patient biology. Do you see this model becoming the future of cell therapy, or could this strategy be applied beyond cardiovascular disease?
There are lessons that can be learned from CardiAMP cell therapy, particularly for autologous cell therapies, which have the great advantage of safety. Pre-screening of patients based on the source cells for therapies makes sense, and in our case, preserves the clinical value of an autologous therapy, while enabling the cost to be lower than a simple allogeneic therapy. Most of the field seeks to abandon autologous cell therapy because of its enormous cost, to focus on allogeneic cell therapies. But CardiAMP presents an approach where the cost can be incredibly low to society relative to other cell therapies. By way of example, I've been on panels where there is a discussion of autologous cell therapy, allogeneic cell therapy, and then CardiAMP cell therapy, as it does present, a new approach for CardiAMP cell therapy. So I think just being in that dialogue can be instructive to other therapies, and it creates, a new paradigm, if you will.
Yeah, certainly, and certainly at a time when cost of therapy is of great concern. So perfect timing for that. CardiAMP doesn't aim to regenerate heart muscle directly, as I understand it. And so can you explain why improving microvascular function and reducing fibrosis is in fact an effective approach to treating heart failure?
Erin, absolutely. So at BioCardia, we are focused on cardiomyopathies that have resulted from poor blood perfusion to the tissue, that are ischemic in nature. And that is ischemic heart failure and chronic myocardial ischemia. Enhancing the microvascular function makes sense in these tissues. The disease derived from poor perfusion on a macroscopic level, but in the microvascular setting, there can be issues as well. And not only do the cells we deliver release angiogenic cytokines, such as vascular endothelial growth factor, to the surrounding tissue, but the therapy actually contains the CD34 endothelial progenitor cell population, which is involved in the growth of new vessels. And in our clinical studies, we have seen benefits which we believe are a result of these mechanisms of action.
Good. Okay. And speaking of your studies, I want to talk a little bit about that. What evidence from the CardiAMP HF trial supports the idea that stimulating capillary growth and reducing fibrosis leads to better heart function? And on top of that, how do the findings set CardiAMP apart from previous cardiac cell therapies that have struggled to show efficacy?
I'm happy to try and address this, Erin. So heart function manifests itself in many ways. So in this trial, when we delivered cell therapy on top of guideline-directed heart failure medication, the data trends show patients have greater survival, fewer cardiac events and rehospitalizations, fewer arrhythmias, modest enhancements in heart function, and meaningfully better quality of life. Pre-specified outcome measures based on these trends have actually achieved statistical significance in patients with elevated markers of heart failure, which we're thrilled with. So we view these results as an excellent phase 2 trial, perhaps the best large trial of a cardiac cell therapy ever reported, but recognize that additional evidence of both safety and efficacy have value. We also believe our proprietary elements contributed to these positive results. A precision medicine approach where we pre-screen for likely responders, the high cell dosage that we're delivering, and the precise and safe delivery system in our Helix transendocardial biotherapeutic delivery platform. And so we think these advantages are different from what's gone on before, but at the same time, what we've done has built on all of the work that's gone on before as well. So we've benefited from those past studies.
Great. Okay. You touched on this a little bit at the top of our call, but I'd like to get into a little bit more, a little bit deeper. Given the limitations of traditional heart failure treatments, where do you see CardiAMP fitting into the broader heart failure treatment landscape? And could this approach perhaps redefine how we think about treating advanced heart failure patients?
That's the $1 million question, Erin. So based on results from this trial, CardiAMP has potential to be a minimally invasive and cost-effective adjunctive treatment to heart failure medications to improve mortality and quality of life and reduce cardiac events and rehospitalizations. Initially, based on the data we now have, it should be limited to patients who have been stabilized on guideline-directed medical therapy and still have elevated biomarkers of heart stress. The benefits we have seen in the recent CardiAMP heart failure study are all in addition to those from the other medications the patients are on, not instead of these therapies. The autologous, minimally invasive, one-time treatment nature of the therapy today could make it an attractive option for patients who may need more than current therapies provide. Merely enabling this to be available for these patients is an enormous hurdle that we're working to address. State-of-the-art care for advanced heart failure is continuously and carefully evolving. And if CardiAMP cell therapy is available, it will be part of this evolution, which will be driven by rigorous clinical science. Autologous minimally processed cell therapy, where cell collection, processing, and delivery take place in one bedside procedure, it's likely to be far more cost-effective than allogeneic donor cells that are heavily processed and complex to produce, which we are also working on. And so for that reason alone, it could be, if you will, the frontline cell therapy, assuming there's multiple therapies in the market one day. Because of the cost profile, I think CardiAMP has inherent advantages that will last even as other therapies make it to market.
Excellent. Okay. Finally, we talked through certainly quite a bit about CardiAMP, but in addition, what's on the near-term roadmap for BioCardia, and how can our listeners learn more about what you're working on?
Thank you, Erin. So the main effort right now at BioCardia is we've just completed the CardiAMP HF trial, is we're working on submission of these results to both the FDA and the Japan PMDA, or Pharmaceutical and Medical Device Agency, to explore marketing clearance pathways. The data we have, is good. It's not perfect, but that's a big initiative for us today. In addition, we are undertaking the CardiAMP Heart Failure 2 trial in the patient population that responded well to therapy in the first trial, those with the elevated biomarker for heart failure of NT-proBNP. And we have our other therapies that are coming along as well. So we expect to have top-line data for the roll-in cohort of CardiAMP in chronic myocardial ischemia this month. And we expect the Data Safety Monitoring Board review of the low-dose group in our CardiALLO program, which is also in ischemic heart failure, this month as well. So related to all of these programs is also our Helix biotherapeutic delivery platform. And now with roughly 500 patients treated to date, we will be separately seeking FDA approval of this platform based on this recent large data set. So that's all the near-term initiatives here at BioCardia. If folks are interested in learning more, I would point them to our website, where we do our best to keep folks up to speed. And we, as a publicly traded company, have SEC filings that detail everything per the regulations that are out there.
Excellent. Okay, thank you. And I know that is certainly cardiovascular disease and heart failure is of big concern to our readers of Cell and Gene and listeners to this podcast, especially as we cover non-oncology indications and cardiovascular disease, and all the things we continue to cover is certainly top of mind for our listeners. So I'm sure you'll get a lot of traction from the folks who are listening into this podcast today. And that brings us to the end of this podcast featuring BioCardia's Peter Altman. Peter, thank you so much for your time and your insight. This has been wonderful.
Well, we appreciate the opportunity, Erin. You have a great day today.
Thank you. Thank you. And listeners, please visit cellandgene.com and please subscribe to Cell and Gene, the podcast, so you never miss an episode. And we will talk to you soon.